So we have got our first two presenters for this session will be joining us online. So we'll just go through their bios before we start those videos for everybody. So our first keynote speaker for this session is Dr. Zoe-Joy Newby, who completed her PhD in 2014 in phytopathology, and using her experience in working with fungal organisms, began working with orchids thereafter. For the last 5 years, she's been a member of the germplasm conservation team at Plant Bank, working on orchids as well as rainforest conservation project. Most recently, her work has involved conservation research on several endangered orchid species under the New South Wales Save Our Species initiative. And our second speaker, also online, is Jerry Smith, who is employed as the Threatened Flora Ecologist for the Hills and Fleurieu Landscape Board. He has been working in on-ground plant conservation for over 20 years and is currently delivering a project to improve the trajectory of 20 species of national significance across the Mount Lofty ranges.
Good morning, everyone. I'm speaking to you today from Dharawal country, and I would like to acknowledge and pay my respects to the traditional owners of this land, the elders past, present and emerging. For those of you who don't know me, I'm Zoe-Joy Newby, and I'm a scientific officer at the Australian Institute of Botanical Sciences based at Plant Bank, Mount Annan. And I'm really pleased to be able to talk to you today about some of the research that we've been doing over the past few years in the conservation of terrestrial orchids, particularly looking at some of our challenges, but also the achievements that we have made. So I must take the opportunity to thank the APCC committee for inviting me to speak here today. And hopefully over the next sort of 25 minutes or so, I can give you a bit of an insight into the work that we've been doing here in New South Wales. The Royal Botanic Gardens, as Australia's oldest continuously running scientific organisation, has a long history of discovery and conservation in our native orchids. Obviously, much of this work has been involved with taxonomy of Australian species, but more recent efforts have involved phylogenetic analysis and taxonomic revision. Orchid conservation research has been undertaken by various staff members over the years and included conservation of species such as Diurus, Pterostylis and Caledonia. Many of you will know my colleague Dr. Karen Somerville, who we can see here on the top right, hand-pollinating flowers of Diurus calytrophylla. So Karen has made some significant contributions to orchid conservation research in Australia, and along with the team, a few years ago, Karen demonstrated the ability to re-isolate mycorrhizal fungi from soil samples which had been stored for over a year. Karen and the team also were able to successfully cryopreserve the seeds and suitable symbionts of number of species in sodium alginate beads. And there on the top left, you can see an example of Pterostylis saxicola re-emerging from a sodium alginate bead. The team has also been involved with a number of different propagation and translocation programs. And on the bottom left, we can see Diurus flavescens translocation trial. And on the bottom right, we can see a number of propagated Pterostylis saxicola individuals. Our more recent research has continued to be involved with orchid germination and mycorrhizal requirements. However, we've also done some work looking at germplasm storage, for example, using the cryogenic facilities we have available at Plant Bank. We've also completed a few studies on pollination systems in orchids, as well as looking at population demography and fecundity studies. So as some examples, there on the top left, you can see some orchid mycorrhizal fungi which have been freeze-dried and placed in ampules for storage. On the top right, we can see part of our orchid living collection we maintain at Mount Annan and some species of Pterostylis and Diurus in flower. The lower left image shows a Radimeris wasp, which we now believe is the pollinator of Calocalus pulchellus. And at that lower image is the results of a population study we did a few years ago on Microtus angusii. And in this case, we were looking at the density of individual plants across their type location north of Sydney. Since 2018, I have taken over the majority of our orchid research, working closely with my colleague Jess Waite. So in 2018, we started our first orchid program under the Saving Our Species initiative, and in this case we were looking at 2 midge orchids, Genopleasium plumosum and Genopleasium rhyleticum, which are the first 2 images on the left there, and continuing to move towards the right we were also working with 2 leek orchids, Prasophyllum affine and Prasophyllum petalum. Our second SOS project was looking at the spider orchid Calladenia tessellata, the pretty beard Calochilus pulchellus, the rusty hood Pterostylis dispectans, and Thalimarch cangalunicum, one of the sun orchids. So we're now only working on 6 of these 8 species, but in the current financial year we've also picked up a project looking at the endemic underground orchids of New South Wales, which are Rhizonthelus speciosa and Rhizonthelus latterii. So each of these species is threatened in New South Wales owing to their small population numbers, but they also experience threats possibly from inappropriate fire regimes, habitat destruction, weed invasion, and climate change. This map shows the distribution of the SOS management sites that we are working in. So as you can see, they're spread quite a way across southern New South Wales. Our most distant site from Mount Annan is the population of Pterostylis dispectans at Moama, southwestern New South Wales at the New South Wales-Victoria border. In southeastern New South Wales, down towards Eden, in the area of Pambula and Delegate, we also have 2 more populations, and our most northern populations are at Bulladilla and Barrington Tops, where Rhizontella occurs. So we need to work quite closely with our threatened species officers in DPE and also with New South Wales National Parks to make sure that we can obtain the necessary seed, soil and tissue samples that we require for our research. At the heart of our SOS projects is orchid propagation. We're aiming to get plants in pots, but as some of you may well know, when it comes to orchids, this is definitely no easy process. So as with any plant propagation project, one of the first things you will often require is seed. And this is certainly where the challenges with orchids begin. Beyond that, we also then need to acquire a suitable mycorrhizal fungi. So because orchid seeds are so small, they lack any sufficient form of endosperm and they need to be infected by a fungal organism to allow germination to occur. Once we find that suitable fungal organism, then we need to work towards optimizing our germination processes and getting plants large enough so that they can become independent and we can exflask them and bring them out into the natural environment. But we haven't actually successfully been able to propagate orchids until we can establish them in our nursery. And I think the true indication of establishment is whether or not you have plants re-emerging in a subsequent growth season. Now, for some orchid species, this process of propagation becomes a little bit more complex. And in the case of Rhizontella, these particular orchids exist in nature in what's known as a tripartite relationship. And this means that not only does Rhizontella require a suitable mycorrhizal fungi for germination to occur, but it must also exist with a host plant. So in this case, the first thing that we need to identify is not only a potential mycorrhizal fungi, but we also need to identify a host plant that the fungi interacts with. We can then, once we've established that relationship between those two parts of this interaction, we can then introduce the seeds as the third member of this tripartite relationship. Continuing along, we then will obviously move towards pot trials and hopefully get plants established and see them continuing to emerge and flower in subsequent seasons. So in either case, the process of obtaining seeds is often quite quick if you're able to collect at the right time of year. Getting suitable fungi can be done in a couple of days, but will often take just a few weeks. But then as we move along, processes of germination and exflasking start to take a couple of months, several months. And then as we move into establishment, because we're having to wait to a subsequent season, we're now actually talking about a process of years. So the whole orchid propagation timeline is really quite a long-term commitment. So while obtaining suitable seed is a fairly routine action in most conservation programs, this is often our first challenge in orchid projects. So for particularly working with these SOS species, certainly 2018, drought really hampered our ability to obtain seeds. And it was because we were finding that drought was suppressing population numbers, it was baking seeds before they could completely develop in the field, and it was just reducing overall fecundity within the populations. Pollination failure was possibly also an issue in the field. And I think that maybe genetic issues are hampering some of our species. I don't have any data for this, but given their low population numbers, we know that this can have consequences for seed output. In response to these issues, we generally always use hand pollination where possible. And beyond that, we're looking to bag plants to protect them from herbivory, but also to help collect the seeds, and then also, as necessary, caging as well to keep away the larger herbivores. As has been demonstrated for many orchid species previously, hand pollination can increase seed set. And our colleague Lachlan Wilmot from DPE recently published a paper in 2019 looking specifically at this in Thalia cangalunica. And it was found for this species over a 3-year period, capsule production increased from 4.4 to 23.1%. Which was a great outcome for this species. So hand pollination also increased capsule production in Calochilus pulchellus from 91 to 97%, again looking at a 3-year average. And you might wonder, well, if natural seed set was already quite high in this species, why would you even bother? Well, this particular species is in fact self-compatible, and so we're using hand pollination as an approach to promote cross-pollination, and we know that this has significant benefits for both the quality and quantity of resulting seeds. Hand pollination in Caledonia tessellata has mixed results over the time of the program. Particularly in 2020, we found that although there was about 80% capsule production across the flowering individuals, unfortunately the larger majority of those aborted. I do, however, think that at the time this was a consequence of change in their habitat. So in 2019, our monitoring site for this species in Moreton National Park was actually burned in the summer bushfires. And so the subsequent season, there was much less vegetation on site and plants had less protection and were exposed to higher soil and air temperatures. And I think that this just created a stressful environment, thus resulting in seed abortion. So, again, once we've hand pollinated, it's really important to try to protect our plants if predation is likely to occur. And so looking at this image on the right here, I took this just a few weeks ago at a Genoplesium plumosum site down in the Southern Highlands. And I was going to the site this day to collect seed and was just really disappointed to find so many plants had actually been predated. And so in this image we can see 7 stems that have each been nipped off, and these are just a few in about a 2 metre squared area in which there was probably over 100 stems, and I could literally find a single intact inflorescence and 2 stems that maintained a single capsule. So this just really goes to show that it's incredibly important to protect your plants if you're hoping to actually obtain seeds from them. Once good quality seed is available, the next challenge for orchid propagation is the acquisition of a suitable mycorrhizal fungi. There are several methods available to do this, and I think we've probably used them all. Initially, we did use soil baiting, and this particular approach has the advantage of allowing us to test at any time of the year, even when plants are in their natural dormant period, assuming that you do have seeds available. So when we completed more than 70 tests, but we unfortunately only found this technique to be successful with a few of our species, and it can certainly take many months for the process to occur. The second method we used was in situ baiting, and in this case you take seeds and place them in nylon packets about the size of a tea bag and bury them on site. You exhume the packets several months later, and hopefully what you find inside is some seeds which have been successfully infected and have begun to germinate, from which you then isolate and purify appropriate mycorrhizal fungi. This particular approach has the advantage that it allows you to map out the distribution of your mycorrhizal fungi across your site, and we certainly had grand plans to use the information to sort of plan possible translocation efforts, but again we found this approach only had limited usefulness. The third method that we used, and I think the most widely applied method, is direct tissue isolation. Again, it's certainly quite quick and effective, and we found it useful for a number of our species. It does have a couple of downsides, however, in that you need to acquire your samples from young, actively growing tissues, so it dictates the time of the year when you can make your collection. And this is because this is the point in the flowers, in the plant's life, when fungi are most abundant and actively growing. By the time the plant reaches flowering, it's starting to digest that fungi, and therefore it's much more difficult to obtain. Secondly, unless you are absolutely certain of the identity of the plant, you actually need to wait for it to flower, mark it, and then return the following season to make your tissue isolation. And finally, depending on where the fungi actually occur within the plant tissue, you may actually need to sacrifice 1 or 2 plants to obtain your tissue samples. So for example, for some species of Hardeniana, the fungi are found in the collar region, and so it's quite difficult to successfully remove a section of the collar without actually killing off your plant. So other than this, we've also used cross-compatibility between our species because some orchids are able to utilise different mycorrhizal strains. We were able to take, for example, a fungal isolate from Prasophyllum pedalum and use it to germinate seeds of Prasophyllum athene. However, we didn't actually find that it worked. We have, over the period of the project, tested, I think, in excess of 100 isolates, and again, we've found a few that are successful, and we've applied different methods for each of the species that we've worked with. So in that lower image on the left, you can see a section of Hardeniana tessellata tissue which has been cut open, and all those little cloud-looking things that are in the tissue are the fungal pelotons, that is the fungal growths within the plant cells. And so there on the right, I'm taking that tissue, I'm sort of pulling it apart and excising the fungal pelotons, and then placing them through a series of sterile water dilutions to hopefully remove any contaminants and help us to purify our culture. Other challenges that we have in relation to obtaining suitable mycorrhizal fungi include some species are probably non-culturable. That is, they are obligate on their plant host, and as soon as we try to separate them from their plant host, they simply won't grow. Others have been quite short-lived, and after subculturing them 3 or 4 times, they stop growing, while others have just been incredibly slow. We have, however, had some success in obtaining suitable mycorrhizal fungi, and we now have 2 cultures for Prasophyllum pedalum, Pterostylis dispectans, and Genoplesion plumosum, 1 culture, likely culture for Rhizonthelus latterii, and a morphologically similar isolate from Rhizonthelus speciosa. Quite interestingly, each of these species of fungal culture that we've got is most closely aligning with Serrata basidiums associated with Australian orchids when we've compared our sequences to those available on GenBank. We're still attempting to obtain suitable fungi for Hardeniana tessellata and Thelymarch cangalunica. We were particularly hampered for both of these species in the earlier years of the project with poor quality seed and low seed availability. However, we've been able to use hand pollination to increase seed production. We've now attained good quality seeds. We've got nice, lovely fresh cultures from live plant material. And hopefully now everything will come together, and the shoot primordia that we're seeing developing will shortly turn green in the next few weeks. Although we've now got seed and suitable mycorrhizal fungi available for a number of our different species, we're still finding that we're getting some challenges with the germination process. So often it's simply just really slow. We're also finding that we're losing fungal efficacy over time, and sometimes our issues are coming from inappropriate germination conditions, which we still need to optimise. So in response, we'll often introduce our fungal symbiont as close to seeds as possible, or on multiple occasions. We're trying to optimise our collection of seeds so that it coincides well with fungi, and then we'll also trial various temperatures and media types and stratification treatments to improve germination. So on the bottom left there, we can see that we've got Prasophyllum pedalum on our thermogradient plate, and this allowed us to identify the optimal temperature for increasing the speed at which germination occur and the number of seeds that actually germinated. In the middle, we're trialling different types of agar to increase fungal vegetative biomass, and on the right, we've got a symbiotic germination plate, which we've actually inoculated on 3 separate occasions with 5 plugs. So after working with some of these species now for nearly 4 years, we're starting to move towards our larger flasking processes. So our biggest issue so far with the small pilot trials that we've had has been transplant shock and inappropriate conditions. So we're having to again adjust our timing and select plants at a more appropriate age, and then also take multiple steps to decrease humidity, increase the light levels, and expose plants to a greater temperature variation. So as many of you can imagine, within our facilities, this can be quite a logistic challenge. So as I began with our discussion of orchid propagation, the true indication of establishment is whether your plants re-emerge in the following season. And so on the larger image to the right there, you can see a tray of 25 Prasophyllum, Pterostylis dispectans seeds that were potted up in April of last year. And looking at them now 11 months later, unfortunately, plants have only re-emerged in 4 pots. However, there is at least 1 pot that now contains 3 plants, and this is certainly a good indication that tuber multiplication has occurred. We'll shortly be setting up quite a large trial, so Jess Waite has very painstakingly transferred over 2,000 Prasophyllum pedalum seedlings into symbiotic replating medium, and they're now getting large enough that we are about ready to transfer them into potting mix. So we'll be looking at a number of different potting media, such as Akidama and expanded clay balls, and using those to see if we can improve soil moisture retention and drainage and get some better outcomes. Although we've made good germination progress with some of our species, we've had no germination with Calichylis pulchellus or Prasophyllum affine. In the case of Calichylis pulchellus, it's possible that this species actually lives in nature in a tripartite relationship. And we suspect this because all known individuals are occurring within close proximity of eucalyptus trees. I'm unaware of any documented cases of the successful propagation of Calichylis, so it's making it difficult for us to troubleshoot where the issue lies. We do, however, have good quality seed. We've got cultures which have been isolated from living plant tissue, but we still just cannot get germination to occur. We've also recently set up a stratification experiment for Calichylis pulchellus, but even after 2 months, I'm still not seeing any good signs of germination. In the case of Prasophyllum affine, the biggest issue that we're dealing with is poor seed set and then no germination. So there are only a few individuals at the monitoring sites that have been sent here, and they're quite a way away from each other. So it's possible that there's issues with cross-pollination in this species. They may have lost their pollinator. We simply don't know. We've also found since 2018 that conditions in the wild seem to be quite poor. So while inflorescence may be produced, they're often getting baked before the end of the season, and seeds actually have the opportunity to mature. So I must thank Mark Freeston for his advice on this particular genus. He's made quite a number of suggestions, but we have tried using different types of media, different sterilisation protocols, symbiotic and asymbiotic germination. We've even tried sowing seeds with our effective Prasophyllum pedalum cultures, but unfortunately none of these approaches have worked. So this species is definitely a candidate for more research, perhaps a genetic study, but we need to spend more time trying to work out what's going on. So while we're beginning to have some success with the terrestrial orchids, working with Rhizoanthella is just that little bit more complex. And as I mentioned right at the beginning, they exist in nature in a tripartite relationship. So they're not only just dependent on having suitable mycorrhizal fungi, but they also relate with an autotrophic plant host. So Rhizoanthella species are known in Australia as the underground orchids. And as the name suggests, they spend, if not all, the majority of their life underground, and the only reason their flowers or their fruit are ever exposed to the soil surface is perhaps because there's been some runoff or some animals digging around them, or through earthworks, and this was actually the way these orchids were actually first discovered in Western Australia. So they lack any chlorophyll, and therefore they are entirely dependent on other organisms to provide their nutrition, but also for flower fertilisation to occur, as well as seed dispersal. So they're called microheterotrophic species, exist in this tripartite relationship, and this is also evident in other orchid genera, including Coelurhiza, Plantenthera, and Sarcochilus. So for us to be able to successfully propagate Rhizoanthella, we first must identify a likely mycorrhizal fungi, and then identify suitable plant hosts, and make sure that mycorrhizae do actually form between our host plant and our fungi. And once we've made that relationship happen, then we can begin to introduce seeds as the third member of this equation. Fortunately for us, the relationship between Rhizonthala and a suitable plant host has actually already been established. So Walkup in 1985 was able to demonstrate that the Western Australian species Rhizonthala gardnerii germinated in the presence of Melaleuca uncinata. Now this was fairly straightforward given that Rhizonthala gardnerii grows basically exclusively with Melaleuca uncinata monocultures. However, our species from New South Wales grow with quite a wide range of potential plant hosts. So again, working with our threatened species officers, we identified 14 likely plant hosts, and so far we've tested 6 of these, and then also included Melaleuca uncinata as a 7th, just given it's been shown to be effective on a number of occasions in the past. So to do this, we took seeds of our potential plant hosts and combined it with fungi, and then 16 weeks later we've had a look at their root systems to see if there is any evidence of mycorrhizal formation. So in the case of both of the Melaleuca species we've worked with, looking at these lower images, we can see in their root systems that they've got these short, stumpy brown sections that are quite a bit thicker and wider than uninfected roots, and they lack any root hairs. Now, I haven't had a chance yet to actually dissect some of these and see if fungi are indeed growing in amongst the cells, but certainly at a gross morphology level, these are good indications that a mycorrhizal relationship has been established. And so the next step here for us is to move on to pot trials, take these Melaleuca species, our likely fungal organism, and then include Rhizonthala seeds. So considering all the species that we've worked with, we've learned a couple of things along the way. It's really important that we get our seed collection and fungal isolation timing right so that we can optimise germination. The different isolation techniques that we've used have been effective for different species. So soil baiting work for Prasophyllum petalum, in situ baiting work for Genoplesion plumosum. We obtained cultures for Rhizonthala slaterii from direct tissue isolation. And in the case of Pterostylis dispectans, we're still getting the best germination with cultures derived from Pterostylis gibosa and Pterostylis saxicola. It's also really important for us to continue to optimise surface sterilisation methods for each species individually. Ideally, as we come to the end of these SOS projects, we should have made some significant contributions to the conservation of each of the species we're working with. And in some cases, we've already been able to meet some of our targets. So as examples, we now have seed banks for each species and fungi where possible. We've got a greater ability and understanding of how to propagate orchid species from New South Wales. We have increased plant numbers held in our living collection, and hopefully some of these plants will actually be used in translocation efforts in the future. And obviously we continue to work towards improving the survival rates of seeds that we raise and plants that we propagate. So while Jess and I still have a lot to learn about orchid propagation, the scientific research team at the Australian Institute of Botanical Science is certainly ready to help you with your orchid project or certainly the conservation of any plant species. So we can help you with identification, population genetics, ecology, pollination studies, germplasm collection and storage, germination, propagation and translocation. And if you do want more information about conservation of orchids particularly, the Germplasm Guidelines Chapter 12 is a great place to start. So thank you to the many wonderful collaborators we've worked with, staff from the gardens and also staff from DPE and others that have made contributions over the last few years. And also thank you to our funding bodies, the New South Wales Saving Our Species Program and funding coming through the New South Wales Government Environment Trust. So thank you for listening, and please do get in touch if you've got any information or would like to talk about orchids.
Hi everybody, hope you're having a fantastic conference so far. I'm going to be talking to you today about the Back from the Brink project and specifically the conservation of the critically endangered Prasophyllum murfetii, the Fleurieu Leek orchid, which is a species that's found within the Fleurieu Peninsula swamps, a threatened ecological community. Back from the Brink is a fairly large project dealing with 35 matters of national environmental significance, a number of which include fauna species and of course one threatened ecological community, the swamps of the Fleurieu Peninsula. The components I'm working on are to do with 20 nationally listed flora species, and the outcomes are that we need improved trajectories for those species against existing baselines, and luckily we do have some good baseline data for pretty much all of those species going back to the early 2000s. Back from the Brink is operating over 80 project locations, so there's quite a lot to organise, and it's to undertake interventions, and those interventions are both looking after the wild populations, the in situ populations, but also ex situ conservation actions like seed banking and propagation of those plants ready for translocation. I wanted to start by talking to you about a sheep paddock, and a sheep paddock's not something you would normally associate with a plant conservation program. But this particular sheep paddock is important because it started to change the way we thought about managing this species. We had visited this site because right next door to it, you can see on the left-hand side, is a fence, and that had fenced off a Fleurieu Peninsula swamp, the Threatened Ecological Community. And there had been a significant program of fencing off swamps from stock grazing over the last 20 years. We visited that site because Denzel Murfetii, who's pictured in this photo there, he's crouching down pointing at the Prasophyllum murfetii, and he's the person after whom this plant is named, had told us he had seen this plant here back in the early 1990s. So we visited the site to look in the swamp for this plant. But we couldn't find it, largely because we couldn't even penetrate into the swamp, it was that overgrown. But on the way back to the car, feeling a little dejected because it was another site where we hadn't found this plant, Denzel tapped me on the shoulder and said, "What's that?" And there on the ground was Prasophyllum murfetii. And we were very excited by that, and we had a look around and found 30 flowering plants, and it was the first new population that had been seen for quite some time. That site now has over 250 flowering individuals. But it did start to change the way we thought about things because we could see that disturbance was required for the flowering of this individual, of this species. Here we can see the location of the sheep paddock, standing with the property owner and me on the right-hand side, and in the background is the Fleurieu Peninsula swamp. And you can see that it is quite thick and you can't even get through that vegetation. Where the critically endangered orchid occurred was outside of that swamp vegetation in the adjacent sheep paddock, which had had sheep in it 6 weeks prior. And this is another site where we found Prasophyllum murfetii. It had been one flowering individual, and we removed that thicket of tea tree swamp and coral fern that you can see in the background behind the fence, and it grew from one flowering individual to over 150 flowering individuals. Now, we're not sure that was recruitment. We think they were tubers under the ground, but the flowering response was in a result of the disturbance that had occurred. So Prasophyllum murfetii was first described in 2000 from a specimen that Denzel sent to David Jones. It had previously been included in the Prasophyllum frenchii group. Prasophyllum murfetii is restricted to the Fleurieu Peninsula in South Australia and occurs in a threatened ecological community, the swamps of the Fleurieu Peninsula, which of course have their own historic land use issues such as clearance and grazing, and so that's the reason they are a threatened ecological community. Here you can see a map showing the distribution of the species. Up in this corner here we have the location of Adelaide in South Australia, and then the Mount Lofty Ranges and the lower lakes, and the area of the Mount Lofty Ranges that it's restricted to is right down on the deep southwest of the Fleurieu Peninsula. So not a lot is really known about the ecology of the Prasophyllum murfetii. The pollinators were certainly unknown, although we did observe quite good natural pollination levels. And out with Denzel one day, we were lucky enough to observe this guy going between the flowers, and Denzel managed to get some photos of it. So we do know that natural pollination of the Prasophyllum murfetii occurs. There have historically been very few collections of Prasophyllum murfetii. Some collections of Prasophyllum that were dated as Prasophyllum frenchii have since come across to murfetii, but it's given us a very limited basis to follow up historic records. In 2017, Prasophyllum murfetii was known reliably from only one location and very few flowering individuals at that location. It was located within a conservation park just outside of Mount Compass on the Fleurieu Peninsula. So some of the things that we've been doing from back from the brink is to actually undertake some interventions, and one of the key ones of that was we made a presumption that it would be easier to conserve populations where they occurred than try to propagate them and plant them back. And certainly we didn't feel as though we had a good understanding of all of the locations where Prasophyllum murfetii might occur on the Fleurieu Peninsula. So we did start to look at the limited set of historic records and have tried to follow up on going and visiting all of those locations. Now, we did find plants at some of those historic locations, and what it allowed us to do was refine and expand the types of habitats we searched. And there were three main vegetation types we looked at. One was the actual swamps of the Fleurieu Peninsula in the wet areas where the peat occurred, and very few plants occur in this community. Where they do occur, they're normally on the top of a mound within the swamp where they're not completely wet. We only really saw this plant in that sort of location at one site. We also then looked at the edges of the swamps where it comes out of the silky tea tree, which occurs with permanently wet feet, into the prickly tea tree community on the edge of swamps with lepidosperma. And probably the majority of the populations occur in that vegetation type. And then the third type was something that was fortuitous for us in a spring wildfire. Went through an area of swamp gum woodland, Eucalyptus ovata woodland, and we noticed some leaves of Prasophyllum murfetii in that habitat as well. So we started to expand out a little bit in terms of where we looked for this species. As we searched for the species and found it, we have started to use a field-based mapping program so that we can get polygons around the areas where they occur, so that we know that we can go back and compare over the same area. Previously, all the data was point-based, and so we really didn't have much idea of what the extent of those populations were. It would just have a GPS coordinate and say 45 plants, for example. So we really wanted to have some sort of mapping approach, so we've brought that onto a field-based tablet approach. And the result of this was that through all of this effort, 5 new locations were discovered, and a lot of those locations where we'd done some other actions, we started to see really good populations. One of the other things we've been doing for the conservation of Prasophyllum murfetii is habitat augmentation, acknowledging that most of the locations had some previous and historic disturbance. So at Stipaturus Conservation Park, an ecological burn was undertaken with the DW fire team. The result was an increase from 3 flowering plants to about 30 flowering plants. This is where it occurs. It still leaves quite a bit of the canopy intact. If you're wondering what that path is through there, that's the Orchid Enthusiast Superhighway. This site certainly gets a lot of visitation. The other thing we did, this is another location where Prasophyllum murfetii occurred, it was down to 1 flowering individual, is manual slashing and the thatch removal. And you can see one of the people doing this work here is particularly impressed at the prospect of this hard physical work, but unfortunately it was the only way it could be done. If you're worried about that clearance impact, and it was approved clearance, you can see here how quickly it has grown back. That's 6 months later at the same location. But this was really successful, and this site went from 1 flowering individual to over 150 flowering individuals within the space of 18 months. And so the final thing we're doing for the conservation of this species is undertaking ex situ conservation, really as a backup and to complement what we're doing. We did manage to get some really good seeds banked. Over 7 million seeds are now banked, and here you can see Ty from the South Australian Seed Conservation Centre posing for me fantastically, looking at the seed capsules on a plant. And there's some of the seed from Stipaturus Conservation Park. The other thing we've started to do is in vitro propagation and some of the fungal isolation. This has been done both at the South Australian Seed Conservation Centre and in conjunction with Nushka Reuter over at the Royal Botanic Gardens of Victoria. And there has been some really good success. And here you can see some of the young Prasophyllum murfetii on agar plates over at the Botanic Gardens in Victoria. So what's the future of this? Well, obviously all of these lovely propagated plants need somewhere to go, so we do plan to do some in situ translocations. Most of the locations now are fenced from external grazing pressure, and they present a fantastic opportunity to bolster those populations. We would like to continue the ex situ breeding programs. We think a backup population is critical for this species, and it certainly makes seed collection much easier. We still have some historic locations we'd like to continue searching for. We don't think that we've got to all of them yet, and we just need to keep assessing our management options and the opportunity interventions. We're not entirely sure that we've got the habitat augmentation component right yet, or certainly at the right intervals. So there's still a lot to go, but so far the signs are really good. We've gone from 3 flowering plants to over 700 flowering plants, and from 1 reliable population to over 7 populations. So the future's looking bright, and we hope we can continue this momentum into the future.
So according to my timeline, we're a little bit ahead of time, maybe about 5 minutes, which is, I think, good. So we'll keep going. So our next speaker is Dr. Samantha Yap. She is part of the Research Centre for Ecosystem Resilience at the Royal Botanical Gardens and Domain Trust in Sydney. And she's leading a series of conservation genomic projects across a wide range and a number of threatened species. And that's to guide conservation efforts that will help secure species survival. Thank you, Sam.
Hi, today, switching up a bit from all the orchid work, I'll be following on my colleagues from this morning about threatened species, other threatened species, and also talking more about genomics and sowing seeds for opportunities of using genomic work for the future, and it's all very hopeful. So my name is Samantha Yap. I'm from the Royal Botanic Gardens, Sydney, the central button. Okay, thank you. Yep, and based on yesterday's talk by Maurizio, you all would have known that we come from the Research Centre of Ecosystem Resilience, which is part of the Royal Botanic Gardens, Sydney, and it was touched on that we have established large-scale data generating resources and also a network of people that we know of, and that has enabled us to generate genomic data both at a timely manner and also at a cost-effective manner. And with that genomic data, we combine it with climatic, environmental, ecological data sets that enable us to provide very informative solutions to help with the preservation and restoration of plant diversity. The bulk of my work, of course, is on one of the workflows that we have produced over the last few years, and this workflow has been really robust, has been worked on at least 40 threatened species, that some of which was mentioned this morning, and it's part of the Saving Our Species initiative by the Department of Environment and Planning. And so with that, you can read more about this workflow that we have generated in order to work through all these threatened species that we were able to get through in the last few years. But that's made possible because we have a one-stop shop sampling strategy that allows us to address study questions that are relevant to what the SOS officers need, and in that way also contribute to applied outcomes associated with species management. And a bit more about a case study now in order to show you just how useful this workflow has been to us. It's on this enigmatic species called Eidothea hardeniana. It's not as well known or as rare as the Wollemi pine, but don't fall asleep just yet. Its recovery was still quite a surprise to all of us because it was discovered in the Nightcap National Park, where it was quite a well-studied and well-surveyed region. And it was discovered by Rob Koyman, of course, who's really the expert in that area. And he also later described the species with Peter Weston. This species belongs to Proteaceae. It doesn't look like that from the pictures here. It's not as showy as other Proteaceae, but there is evidence, of course, that it belongs to Proteaceae because there's even fossil seeds that resemble this species that indicates that it's quite a basal lineage. So because of all that interest, naturally the conservation was well attended to, in that there were was extensive surveys being conducted, and all of the known individuals of Eidothea hardeniana were tagged, and we were after as much information as we can get from the plants itself. And also there was a genetic study on it right after its discovery, which was within the last 20 years. But more recently, there's motivation to update that information about species genetic health using the latest genomic technology. And this is where the SOS officer, Justin Melly, came in. He was really keen to apply genomics to understand more about the species. So he funded this project and assigned Louis Weber, who's also another rainforest expert, to collect all the leaf tissue samples for this study. And I should mention also that a lot of the SOS projects that we have been involved in were funded by the SOS officers because they were really keen to first use the genomic data in order to understand how to apply that for conservation first. Previously, it was always the other way around, where we do all the on-ground work first and then genomics. So yeah, that's where we are at. The sampling started in 2019, but as with many parts of Australia, the rainforests in Nightcap National Park were also affected by the 2019 to 2020 bushfires. And the rainforests there were impacted by fire, even though there was barely any history of fire affecting that place previously, and let alone this species, which we did not know whether it will survive the fire. Thankfully, it did regenerate. Most of the plants managed to resprout, as you can see in the image. So sampling was well underway by then, and we were able to include some individuals that we now know have perished. This is quite meaningful because we are able to use genomic data to tell whether these individuals have unique diversity that is permanently lost as a result of the fire. Oops. Because of the fire, sampling was also delayed, but also that we were able to include the ex situ collection that was established as an insurance population, because we were unsure at the time whether the species was able to bounce back again after, say, if another fire happens. So this collection consists of seedlings that were germinated from in situ seeds. And these seeds were collected off the base of the trees around the area. And so what we know from previous observations was that the seed dispersal is limited because the seed itself is not very palatable to the local dispersers. That's why most likely dispersal is limited. And so, yeah, we were able to record down the putative mother plants for all the seedlings that germinated. And yeah, in the map that is shown there, we were able to plot down where all these seedlings came from. Oh yeah, and also this collection was included in the sampling so that we could understand how much diversity is being represented in that collection, because we really want to ensure that it's long-term viable, so that, yeah, we can ensure that the species can survive even though the in situ population might not. So once all the time-consuming sampling was done, it's up to us now with the leaf tissues that were obtained. We pack all the tissues into tubes and then send it off to a company that does DNA extractions and sequencing. And the whole process was it takes 2 months for them to return with the data, and then we do all the writing and analyzing, and the results that came out of that took 3 months in total. So all in all, if a project has an even more straightforward sampling strategy, we can actually get all the results within 6 months. So that's quite a lucrative idea to include genomics, if you think about it, yeah. But in this case, Eidothea, it wasn't the case. But then the study questions that we can address using the genomic data is quite exciting. And here's what we found. So as I mentioned, the sequencing company returns the data after 2 months. It's in the form of a spreadsheet, surprisingly. So this spreadsheet contains single nucleotide polymorphisms or variable sites, and that's like thousands of them across the genome in order for us to look at variation across all the individuals that are grouped into populations. And it's even very informative for looking at species level differences. And yeah, so the first thing we do when we are given the spreadsheet is to run a PCA analysis, principal component analysis, which tells you variation on a multivariate space. From the results here, we can see that individuals are not really distinctly clustered. So you can, based on this, we can say that the species can be treated as one single population. And yeah, we highlighted the individuals that have died as a result of the fire, and we can see that they are not really distinct from the living ones, which are the colored ones, blue, red, and green. So we can say that the impact of the fire was not that severe, but yeah, it could still be that we have lost a certain amount of diversity. To verify that finding, we also ran a kinship analysis, which is to look at genetic relatedness among all the individuals. So as I mentioned before, dispersal was limited, which means that it's possible that the population has lots of individuals that come from seed, just drop from the ground, and everyone is related to each other. That's why we were keen to find out more about relatedness. Turns out that all the known dead individuals are related to the living, but most interestingly, we find that almost all individuals belong to 4 major families from the main subpopulations, or a large proportion of the population. And what I define here as a family is that individuals had at least full sibling relationship with another individual, which is quite related. So now that we know that everything's quite related, it is really important to understand how much diversity was captured in the ex situ collection. And even without having that genomic data on the collection, based on this map itself, I've colored all the maternal parents by which family they belong to. You can tell that the collection isn't capturing representative diversity of the whole species. And with the genomic data of the ex situ individuals included from this principal component analysis plot, we can see that, yes, it's really not representative of the diversity of the overall species. So what can we do about that, and how do we improve on that collection? This is where the work of Jason Bragg comes in. He has developed a very neat analysis of optimally select individuals for any given propagation population size, and propagation populations that can be used for ex situ collections or translocation populations. So for the case of Eidothea hardeniana, we ran through this analysis to find out what other individuals in situ are needed to supplement this ex situ collection in order to boost the representation of diversity. And it's in the form of a spreadsheet with the specific individuals to select in pink. And also, we provide additional scenarios so that it's flexible to just about anyone, even if there are limitations at the nursery where you can only hold a certain amount of individuals, then you might only want to include a few more. And that's what this plot is saying, is that with the square dot, is saying that that's the amount of diversity represented by the ex situ seedlings only. And if you include 6 more in situ individuals, you can actually boost the species, the amount of diversity represented by the collection. And from this plot alone, we also see that how much diversity is represented if you randomly select the individuals for that collection versus if you genomically select them. You can see that they are very similar as you reach a population size that is larger, and that's quite interesting because sometimes if you cannot, you don't know the identity of the individuals, it would be best to spread out that collection. Okay, so from this body of work, we have learned quite a lot about species biology, ecology, and also provide a lot of applied recommendations on how to improve on the ex situ collection. But also we found that we cannot rely on just the seed alone for the conservation of this species, and that in future we need to look at collecting the seeds as part of the active management of this species in order to avoid the possibility of seedlings inbreeding with the adults. And so overall, the workflow that I've presented and this case study, we have found that there's quite a lot of information that we can gather through genomic data. And all the threatened species would have unique problems that are associated with them. But then with genomic data, as well as working with a lot of the landholders and people that are expert with the species, we can actually learn a lot. Thank you. Oh, and I would like to acknowledge Justin and Louis' contribution. And we have a workshop in May that anyone can feel free to attend. And it's on conservation and restoration genomics for field practitioners. Thanks.
Thank you, Samantha. That was a really fascinating talk. Really appreciate it. So we're going to move on to 2 presentations which are a mix of online and a little bit of oral to begin with. So we're going to start with Dr. Amelia Martyn Yenson, who will first speak for about 5 minutes and then move to a video. So Amelia is currently a project manager at the ANPC and coordinated updates for the 3rd edition of the Plant Germplasm Conservation in Australia. Amelia has a research background investigating seed lifespan and germination, with a focus on improving seed quality for conservation storage of Australian native plant species. She has also a growing interest in science communication, and Amelia's research is based at the Australian Seed Plant Bank. And because we've got these two double-barreled online presentations, we'll quickly introduce Dr. David Bush, who's unfortunately not able to join us, but did send in a video, pre-recorded one. So Dr. David Bush is the Director of the Australian Tree Seed Centre and a forest scientist that has been involved in testing, domestication and genetic improvement of tree species for over 25 years. His research has been focused on selecting Australian tree species for commercial use in Australia and overseas, as well as conservation of forest genetic resources in Australia and in the Pacific. So welcome, Amelia.
Before we begin, I'd like to acknowledge the traditional owners of the land where I live and work, the Darug and the Darawal people, and I pay my respects to their elders, past, present and emerging. And I also acknowledge the Wiradjuri people whose country we gather on today. It's great to have an opportunity to share one aspect of plant germplasm conservation in Australia with you. These germplasm guidelines, as we call them, are the third edition of the Australian Guidelines for Ex Situ Plant Collections. They're a joint project of the ANPC and the Australian Seed Bank Partnership. And the project is funded by the Ian Potter Foundation, and their funding is one of the reasons that the guidelines are available for a free download and also available at low cost from Joe on the registration table. Quick advertisement if you would like a printed copy. The germplasm guidelines are evidence-based, technical and practical, and they give readers an access point into the literature. The guidelines are cited nationally and internationally, and this edition has been downloaded more than 500 times since it was published in September last year. The guidelines provides a workflow or a toolkit for planning, collecting, storing and utilising germplasm. This germplasm, which just means any plant tissue that can regenerate into a living plant, might be stored as seeds in a seed bank, as cuttings or seedlings in the nursery, in tissue culture, or as cryopreserved collections stored under liquid nitrogen. These collections are stored ex situ or off-site, so they're available to provide information and plant material to support in situ or on-site populations. And we saw this in Nathan's keynote this morning about Petunia. Although this session today is all about threatened species, in the guidelines we talk about prioritising species that exhibit endangerment, endemism, evolutionary uniqueness, ecological potential, economic value, ethnobotanical or cultural value, and emblematic or iconic status. So they may be threatened or at-risk species or more common species. We've come a long way in understanding and documenting the requirements for plant species stored ex situ in the 12 years since the previous edition. So what we aim to do now and into the future with guidelines such as these is make sure that we've documented and shared effective ex situ conservation for the widest possible range of species. As an example, the previous edition includes 65 genera by name in the index, and this edition includes 116 genera. The previous edition mentioned 25 families in the index, and this edition includes 117. Capturing information on this scale is really only possible by working closely with research, seed bank and horticulture staff around Australia and collaborators in other institutions who are banking and working on wild species. As an example, we had 78 contributors for this edition and we had a peer review process before the guidelines were published, and Susan was one of our reviewers. For each ex situ collection made, it's important to consider the end use of the collection and how healthy plants will be regenerated from those collections and returned to the landscape. Conservation translocation relies on seeds and nursery-grown plants from ex situ collections to augment small and declining populations. This is usually part of a broader recovery plan to prevent species extinction and loss of biodiversity. It's important to consider the intended end use when planning and undertaking germplasm collection. In the video to follow, these considerations are presented as part of a case study on Grevillea caliantha in the Proteaceae family, a species listed as critically endangered in Western Australia and endangered under the EPBC Act. A variety of other end uses for ex situ collections are outlined. And as we've heard throughout this conference, the importance of partnerships, such as with the Australian Seed Bank Partnership for achieving conservation goals, is highlighted. Enjoy this video, put together by Chantel Doyle and Michael Lawrence Taylor with experts from the Western Australian Seed Centre at Kings Park in Kensington.
Plant germplasm is any living collection of plant material that represents the genetic line of an individual or species. Globally, collections are increasing in number and diversity. It's important to understand then why ex situ collections are made and how they will be used in the future.
So there are multiple uses for ex situ germplasm. First and foremost is obviously conservation. We can also use it for ecosystem restoration, plant breeding programs, or for horticultural programs. In places like Kings Park, where are botanic gardens, the germplasm can also be used to grow plants for display, which is an important component of public outreach and education. Depending upon why we collect germplasm in the first place will dictate how we collect it in the field and then what kind of material we collect as well. So for example, when we're collecting for conservation programs, we often need to collect maternal lines from individual plants. But then for other end uses, such as plant breeding, we might be interested in particular forms. So plants with different flowering colour or flowering time are of interest.
One of the most frequent conservation end uses is translocation. That is returning plants to the wild, either as new populations or bolstering existing ones. In this video, we are going to learn how an ex situ collection can be used for a conservation translocation.
Translocations are about establishing viable populations of plants in the wild and ones that have the genetic diversity that allows them to adapt and persist when conditions at the translocations change.
The Western Australian Seed Centre have been conserving Grevillea caliantha with the long-term goal of restoring wild populations.
Well, Grevillea caliantha was listed as threatened way back in the mid-90s. So it's known from 6 small populations up from the Dandaragan area, which is about 150, 200 kilometres north of Perth. Most of the populations were only known on road verges, and it was challenging to manage these populations in the long term. So translocations then became one of the most useful recovery actions we could do to make sure that this species had a chance of actually persisting in the long term in the wild.
Before commencement, the team needed to develop a translocation plan, which detailed the how and the why of the project. They also needed to review permits, approvals, hygiene risks, and exit strategies. Then they could start wild seed collections based on their decided sampling strategy.
So for a conservation seed collection, we want to make sure that we sample widely both within a population, but we also want to make sure that when we're sampling a species that we sample across the populations. And we want to make sure that that sampling is random. But for other purposes, maybe for plant breeding, you might want to make considered decisions when you're selecting for forms or flower types or those sorts of things. If you have populations that are quite disjunct, you then need to make a decision about whether you can mix those populations in your translocation. In the case of Grevillea caliantha, all of the populations occur in a relatively small area. It was decided that all of those populations could be mixed when a translocation is undertaken.
Once in storage, propagation methods can be tested and include vegetative or seed germination.
For rare species, often we don't have a lot of seed to be able to try lots of treatments in order to work out the optimum way of germinating something. So we might use surrogate species to work out our methods and then apply it to our threatened species so that we're not using up that limited seed source.
Trying to establish how best to germinate or propagate Grevillea caliantha was done through trial and error and also using background information that people already knew about how to germinate or how to propagate Grevilleas in general and other species that also occurred in the same family as well.
Once the living collection is established, reintroductions or translocations can occur, but that doesn't mean the extra collection is no longer required. Often an experimental or staged approach is needed.
We've been trying different methods of establishment as we've gone. We don't go too large at any one step of the process, so that if for some reason things don't go right, we still have more seed and opportunities to come back and repeat that process in the future.
So after we had planted them, we installed a watering system to make sure that the plants could be watered over the first 1 or 2 summers. And then also we installed fences to ensure that they didn't get grazed by herbivores such as kangaroos and rabbits. The other important thing that we do when we put plants out there is we tag them. And we ensure that we record all the information that we already know about those individual plants, and then we can follow each individual plant at the translocation site. So we have actually had quite a reasonable number of plants survive, and they've produced flowers and seed. We've had very limited recruitment. We know that recruitment is probably not likely to happen in large numbers in the absence of fire. Some sites are doing better than others, so as we learn which sites are better for establishing grevilleas, we're focusing more and more on those sites. And then we're thinking about, well, what are our next steps as well? So success comes in many forms. It's not just understanding whether the plants survive, but it's also other aspects of their ecology and their life cycle.
Conservation collections are a long-term commitment and need replenishing. Often, fresh collections will be sourced from the wild, but it isn't always an option.
In the case of Grevillea caliantha, some of our seed collections represent populations of the species that are now extinct in the wild. So in that instance, regeneration is really the only option. The next step in that translocation process will actually be to establish a seed orchard to bulk up seed of that species. But regeneration can pose problems for a long-lived perennial species that may take a long time to reach maturity or may require a very large area to grow an adequate number of plants. So in those instances, we would then try cuttings. So some plants can be successfully grown from cuttings. We can still get a good genetically representative sample to use for those translocations.
Translocation is one potential end use. The decisions at project commencement about intended use will inform everything that follow, including collection, propagation and storage methods. But there are always unexpected challenges and uses, which is why partnerships are so important.
Partnerships and collaborations are absolutely essential for starting and maintaining ex situ collections. Conservation often doesn't receive a lot of funding, and we require support from public and private corporations to support the work that we do. Organisations such as the Australian Seed Bank Partnership, which really put together a coordinated Australia-wide response to ex situ conservation, and ensuring then we also have the duplication of material across Australia. And then finally, for research using ex situ collections is often multidisciplinary and requires a great deal of expertise across a wide range of different areas. So including plant biologists, soil biologists, or even people who work with animals.
Detailed information about utilisation of ex situ germplasm is included in Chapter 15 of the Guidelines. The FloraBank Guidelines deal specifically with seed sourcing and ecological restoration. The Translocation Guidelines can be used to plan and undertake best practice translocation.
Hi, my name's David Bush. I'm the Director of the CSIRO Australian Tree Seed Centre. I'm based in Canberra. Talking today about opportunities for growing native tree species for bioenergy production and carbon capture. There are some opportunities to include threatened species in that activity. I'm involved in a project with New South Wales Department of Primary Industry. You can see my my collaborator there is Fabiano, and he's the leader of a project which is entitled Biomass for Bioenergy, and several of the slides I'm showing have been developed by his team there. Okay, this slide just shows very quickly how biomass for bioenergy actually works in terms of the carbon cycle. You can see there it's a forestry example. Forestry plantations are grown, they're sawn up into products in the sawmill, which are used to build houses. You can put wood direct from the forest, or residuals from the sawmill, or wood waste from the demolition of those houses when they've finished their lifespan, and they can go to make heat and electrical power. They can even be converted to liquid biofuels. On the face of it, that doesn't seem too good because that emits carbon dioxide, but of course the forests soak those back up again, and so that's a cycle different from burning fossil fuels, where you're just taking stuff out of the ground and it's going up and not being put back. This slide just shows the form that woody biomass takes. It's converted into these pellets. The plantation's harvested, chipped. Those chips are dried down and ground. They're compressed, so into a very sort of dense little pellet, a couple of centimetres long, easy to transport and store, and they can even be co-fired with things like coal. You can see there in that example, it's a 25 to 35 years growing cycle, but we're looking at much faster cycles than that, of sort of 2 to 4 years. So growing tree crops for bioenergy does have some good benefits. We've got an emphasis on native species, and especially ones that can be perpetually harvested, so that the shoots, the above ground matter is cut off, but the roots remain intact and continue to sequester carbon underground. We're targeting farmland, cleared land, especially marginal land or marginal parts of farms. Sites for biomass, bioenergy need to be close to where they'll be burnt, so not too remote or near to some capacity. But there's a very good potential to establish a diverse range of species, including threatened species. Some other benefits there you can see on the slide are things like habitat provision, land rehabilitation, shelter for stock, and so on, soil erosion, water quality, all of those things. Now, the same species that are used for biomass bioenergy generation are probably going to be pretty suitable for just planting for carbon sequestration, in which case you plant them and leave them. It's still important that they're able to survive, and they're very tough. They've got to be able to survive drought and fire. A species that's often used is Eucalyptus polybractea, a mallee species, blue mallee in southern Australia. But it's probably not the only one that can be used, and so some of the research we've been doing is looking for alternatives to that species. This slide shows the range of sites that we've established in the New South Wales Biomass for Bioenergy project. There are 11 of them, mostly inland. We've established 15 native species, including both mallees and tall trees. They're mostly eucalypts because they coppice well. There are a few other things as well. 4 of the species are actually threatened. 2 that have sort of already been tested and known to be good for cultivation, and 2 completely untested mallees from the Hunter region. This slide just actually goes into what the species actually are. The A species, A. saligna, A. dealbata, are acacias. There's Casuarina glauca, and the rest are all eucalypts. You can see there we've got the 4 threatened species: Eucalyptus benthami, which is endangered under the New South Wales Act, Eucalyptus castrensis and Eucalyptus pumila, which are the Hunter mallees, and Eucalyptus infera, which is from southeast Queensland. The X's just show which species are on which sites. We haven't got everything on every site because the sites differ. Some are waterlogged, some are very dry, some are a bit wetter. Some ones like Glen Innes are very cold. We've just put the ones that we think will do well. We've got good statistical power here because we've got a range of overlap of species on each of the sites, or all of the sites. I mentioned before that we already had tested Eucalyptus infera. That was part of an earlier project where we were looking for subtropical and tropical mallees. You can see in the central map there, we tested a whole range of species, including from Central Australia, but they're all mallees and grow on tough sites inland. Anyway, that species uncovered Eucalyptus infera, which is not only a good species in terms of growth. We found out that it's tolerant of waterlogging, and it also has a good eucalyptus oil yield. We had one of the trial sites was a farm casino, and the farmer there started growing commercially for oil production immediately after the trial finished. So that's good. If you want to read more about that, you can see the publication there. The other well-tested species is Eucalyptus benthami, very suitable for cold sites. It's probably the world's 10th most planted eucalypt, grown in the US, Brazil, Uruguay and China. Subtropical latitudes, but places that get frosts from time to time. Its survival at Glen Innes was reasonably good, managed to survive the frost there even when other things didn't. This slide shows how at least a couple of the threatened species went versus Eucalyptus polybractea, which you remember I mentioned is the sort of the gold standard, the thing that a lot of people plant for these purposes. And they grew very well on quite a few of the sites. Inferrum castrensis outgrew polybractea, or at least equalled it. The other one, Eucalyptus pumila, didn't do so well in the nursery. There's poor germination, which can be an issue with threatened species. It could be one of the reasons it's only got a small distribution. It might be very subject to selfing or have a narrow genetic base. But anyway, having a couple of good successes is you can't ask for much more than that. Okay, well, it's all very well to find species that are good for purposes like biomass and bioenergy, but what happens when people want to plant it at large scale, thousands of hectares? Well, they might go into the native stands and start harvesting the seed from there, potentially causing damage to them and causing maybe even a downward spiral of the actual, what's left of those species. So it's very, very important to have cultivated seed production capacity. You can see here that we have established seed orchards. In the case of Eucalyptus benthami, we started work 20 years ago, which is when we started getting interested in testing that one, both in Australia and overseas. That orchard, and we've got additional orchards, have produced millions of viables. We don't collect seed from the wild, and in fact, that's the wild stands are pretty inaccessible. We've also done the same for the hunter mallees and Eucalyptus infera in the last couple of years. The central slide there, photo there shows a grafted clonal seed orchard of Eucalyptus benthami, where we're now going and just collecting from little non-reproductive pockets along the Nepean River. So we put a fair bit of effort into making sure that people can access cultivated seed rather than relying on wild seed. Just to further reinforce that point, the planted stands of threatened species can be considered to be part of the ex situ population if they're established from a wide genetic base. And this is just quite a nice message I got in the follow-up to the 2019-20 bushfires. This guy from Brazil sort of saying, oh, look, we're very worried about those fires, but if for some reason you've lost Eucalyptus benthami there in Australia, then we could repatriate some material back. Not saying that relying on plantations as the main ex situ conservation is the thing to do. You still need the proper measures in place, but it does help. Well, in the previous slides, I've talked about the benefits that can come from having biomass, bioenergy. There are some other considerations, though. It's only one component, as I've already said, of the renewable energy generation capacity that we should be looking at. There are also quite a few technical challenges involved with developing clays to harvest, transport logistics, and just making sure that there's enough feedstock to actually supply mills or factories that burn it. They do need a guaranteed supply of feedstock, so it's not the sort of thing that can be just done in little bits. It needs a big plantation resource. I'm quite worried about nutrient mining, especially if those pellets include the leaves. You don't just want to burn all the nutrients, especially in Australia. A lot of Australian sites are already pretty low in nutrient status. Another thing is genetic pollution, which is gene flow from those planted stands into something native nearby. If you've got a small remnant population and a large plantation, then the pollen can sort of swamp it, and you might end up with a lot of hybrids, and which often don't perform well, and the remnants might lose their genetic identity. They're all things that need to be considered further when planting natives alongside existing vegetation. So just to sum up, growing native trees for biomass energy and carbon sequestration can help us to a neutral energy generation. That's a good thing. Hardy native species are ideal for this, especially those mallees and things that can withstand being harvested or burnt and droughts. There's plenty of opportunity to integrate threatened species into the plantings, and that's a thing that probably should be encouraged. But it is very important to ensure that cultivated seed production capacity exists so that people don't go in and just mine the seed out of remnant stands. Well, finally, I'd just like to acknowledge some of the team members who've contributed to the projects that I've been describing here. People from New South Wales DPI and people from CSIRO. And thank you all for listening to the presentation.
So what a wonderful range of presentations we've heard from. While I let everybody gather their thoughts, or people online maybe madly type away to put some questions down, I'd really like to thank Zoe and Jerry for their presentations looking at the complexities of orchid propagations, both Samantha Yap and the video that Amelia introduced, dealing with some of the proteaceae. So we had Eidothea from Sam, thank you very much, and the discussions about Foots Grevillea, Grevillea caliantha, being a nice highlight from the video. And David finished on something quite different, I think, than we've heard throughout much of this seminar series, which is quite interesting, that idea of species being used for carbon capture and the possibility of sometimes including threatened species. So he had 4 that he highlighted, but also raised right at those summary slides some of the considerations for the seed sourcing issues and making sure we don't do further damage to threatened species if we're going to use them in that regard. So I'd like to put it to the floor for questions, and also to the folks online. We have some online questions already.
Yeah, this one's for Sam. Do you think the genomic data will give you an idea of an overall ex situ population size to aim for?
That's for sure that we can do that. That's through a conservation genomic study that relies on the one sampling method, one sampling strategy, and you will obtain a genomic data set that you are able to obtain, you're able to analyze and generate a range of scenarios where you have different population sizes, and also we have that information, say for another species, we can even tell which other sites you need to go to in order to generate that population size that is both has maximum diversity, but also with minimum relatedness or kinship. And that's quite important in terms of for Eidothea hardeniana, where we have already an ex situ collection and we wish to supplement that with the cuttings. But one thing I didn't mention in the talk was that cuttings are actually quite difficult to be generated for Eidothea hardeniana, but It's also the case where seeds are also difficult to be germinated, but with that, with the genetic data already present for that collection, as well as the individuals we know should be included, we have been doing a lot of follow-up, just progress updates on different species as well to find out whether those cuttings have been successfully propagated, and if it doesn't, is it still the right number of individuals to include for that propagation population, and also inform about how best to arrange that collection so that if they flower, you want to prevent breeding between all the related individuals. So yeah, in answer to the question, we do have lots of genomic data to help with a range of different scenarios. Yeah.
Any more online questions at the moment? Not at the moment, so we're open to the floor. Got a question?
Hi there. I have a couple of questions for the orchid lovers. I'm wondering if you do any viability testing on those teeny tiny seeds, and if you do you have any tips for me? And also, I'm wondering about your time frames from collection to getting them in the bank, because I'm hearing that that needs to be pretty quick turnaround.
Zoe, we can't hear you. I can see you've took yourself off speaker. No, sorry, not yet. Jerry, would you like to say something while Zoe's just checking a connection there? Oh no, we can't hear you either, Jerry. I think I've just seen someone run along the side, so there might be a little bit of a technical issue our end, and maybe we can come back to that. It's a great question. Is there anybody else from the floor for the moment?
One for David, if I may. David, the large seed production areas that you're developing, are they fully funded, or do you have to support that program to generate that seed? Are you able to support your activities from seed sales for those species, or is that more a developmental process?
Can you hear me?
Yes.
You can? Well, we established those things using funding that's a combination of funding that flows from some of the more commercial type activities that we do. In the case of the eucalyptus benthamite, we put that in really as a conservation stand, but it has generated quite a large amount of money, certainly enough to pay for those other threatened species stands done. So yeah, we've sort of got a good income from some of those, but threatened species stands we put in probably one can actually make some commercial revenue. And that's from forestry companies. They'll pay things like between 2 and even up to 6 cents per viable seed. And for example, last year we sold a couple of million seeds out of that eucalyptus benthamite orchard. So that actually supports us in some of the other conservation activities that we're able to do, which I think is quite a nice thing. But it's not easily achievable by for a lot of people, I think.
Thanks, David. Now, I've just been told that we can give it a shot to go back to Zoe and Jerry for the question about the teeny tiny orchid seed and testing viability.
Okay, that's better.
Fantastic.
Yay. Okay. Yeah, so good question. I have definitely tried, or we've tried at Plant Bank using TZ staining, but as has been the experience of a lot of people, it's not necessarily accurate. I've also tried using fluorescein diacetate staining, and similarly, it requires optimisation for each species on an individual level. So I was actually going to pick your brains and see how you're going. But I definitely think that there's merit in pursuing using TZ staining or other stains for that matter to look at viability in seed collections, particularly when we have ongoing challenges with testing germination via kind of standard methods. So yes, have done it, have found a little bit of success. The staining certainly works, but the interpretation remains challenging.
Jerry, would you like to contribute anything as well?
Yeah, look, I'll just go along with what Zoe said there. In terms of the viability testing, we don't do any actual viability testing of the orchid seed. It's just generally quite difficult to do. But what we do notice is that the viability from large populations, the rigour of the plants that are grown in the laboratory and in the nursery is much better, and they tend to be easier to work with. So that's really what we use as a proxy for viability, and the timeframes for getting it stored is as soon as practical. Obviously, if we're on a field trip, we can't get it into the bank until we get back, but generally, if we're within that day, into the drying room that afternoon, and then into the bank as soon as possible.
Great, thank you. So are there any further questions from the floor? I think everybody's stomach might be getting the better of them. Oh no, there's another question, yep.
Sorry, one more. Along the same lines, is there much known about the storage life of the mycorrhizal fungi, and do you have to look at longevity testing for that as well?
So that would be a question to both Zoe and Jerry again. Did you hear that one?
Jerry looked like he was nodding his head there, so I was going to leave it to him.
Well, I don't have all the answers, but I do know that it's one of the considerations for conservation, is that keeping the fungus alive is just as important as keeping the plant alive. And for ongoing ex situ conservation, I think the guys, they're cycling through replicating that fungus every 3 to 6 months. So if that fungus isn't actively in your program and you want to come back to propagate it, you're going to have to go through the whole isolation process again. It's a considerable task, and it's really overlooked in terms of the funding. I don't actually do any of that work. That's done by our partners at the South Australian Seed Conservation Centre. But yeah, that's a significant component of the work that we do, is keeping that fungal, the fungus alive. Yeah.
I would agree with Jerry entirely. It does take quite a lot of time, and as I mentioned in my talk, that we lose fungal efficacy for some species really quickly, so in less than a year. So our options are to go back and re-isolate. Maybe we've got plants in the nursery which we can utilise, or we have to go back to the wild again. Alternately to that, we've also tried sort of storing our cultures using cryo technologies, but unfortunately in the case, there's not a lot of research in this area, but in the case of orchid mycorrhizal fungi, at least one estimate suggests that 50% of them can't actually be stored cryogenically. So not only are we battling with their really short lifespan, but also the fact that we don't necessarily have many options to store them. So definitely ongoing challenges. And as Jerry mentioned, it does take quite a big chunk of time to get them and keep them clean too.
Do we have any online questions coming through? No? No further ones there? Well, if there's no further hands up from the audience, I've been asked to do a couple of housekeeping things as well. The first I'm going to mention is for anybody who's on field trips tomorrow, please meet here at 8 a.m. Well, actually before 8 a.m. because the buses are going to go at 8 a.m. So if you're on either of the field trips, please make sure you arrive at this venue a little bit before 8. And the second is that our keynote guest and celebrity speaker for the next session has some books available for purchase. They'll be out with all the other information flyers that are available. So once you've got your lunch, if you'd like to go and have a look at the book and consider some purchasing, that would be terrific. Is there anything else we need to let people know? That's everything? Great, I won't hold you from lunch anymore. And thank you very much to all our speakers for this session. It was a really lovely continuation from this morning's session, earlier the morning session as well.